Okay, so this is going to be a brief overview of public data resources in Bioconductor. Now, there are many of these, so this necessarily involves a bit of curation and selection on our part. But I think we're showing not only the work that has come from ourselves, but some additional things based on some of the most widely used and long-standing and stable and we think usable resources in Bioconductor. So this is going to be a fairly broad overview of a number of resources covering microarray data, RNA-seq data, other genomic data, as well as topical areas such as metagenomics, pharmacogenomics, and the Cancer Genome Atlas. So we're going to start out with Sean Davis, who is the creator of a couple of the most widely used data resources in Bioconductor, the Gene Expression Omnibus, which has been one of the most popular packages in Bioconductor for many years now and remains relevant. The package, is called Geoquery and remains relevant for RNA-seq data, as well as the newer package Genomic Data Commons. So we'll start with Sean talking about that, and then we'll go to Ben Haibe-Kains, who has developed a wide range of resources for pharmacogenomics and toxicogenomics. If you work in those areas in Bioconductor, you will be using his lab's work. And then I'll finish off with some work on a variety of curated and meant to be very easy to use resources. So do you want to take over the screen share, Sean?
Sure. The first workshop, so I'm going to give a couple of background slides. So one of the things that Bioconductor is built on, and I think has made it very successful as a project over time, is this core value of reuse and interoperability. So in the center of this slide, we see the summarized experiment. And for those of you who are familiar with summarized experiments, you'll know what I'm talking about. If you're not, then you'll learn it a little bit over the week. But the summarized experiment allows us to contain, to encapsulate all the details associated with a data set of a multidimensional data set, which is typical of biological data. And once we have one of these summarized experiments, wherever it came from, we can use it to do interesting analyses. So the idea here is that the lowest common denominator for a lot of analytical tools and packages becomes one of these smart data containers. And from there, we can branch off to any one of the circles around the outside, plus many more, of course. And so when we're talking about public data reuse, we're often talking about taking public data resources, which are easily accessible and available, and building the glue or the bridge between those public data resources and the data containers that allow us then to jump off into analysis. Bioconductor kind of sits in the center, in our mind, of course, in the genomic or biomedical data ecosystem. And so we're going to be talking about Bioconductor connections to a number of the different data resources available on this page. And when I say that there's a connection between Bioconductor and one of these resources, I mean that there's literally software built to facilitate, make it as simple as possible for users from Bioconductor to access the data in these resources. And again, you can build your own tools to do that kind of thing, but Bioconductor has that middle piece, that the data container, the summarized experiment, or other data containers that allow us to jump off quickly to analysis. I'm going to talk quickly about two packages. Geoquery is the first. NCBI Geo is one of the larger gene expression databases. It includes about 3.5 million public samples, publicly available samples. And in addition to gene expression, things like ChIP-seq are also available there. Until recently, most of the data were available on microarrays, but now there's a, obviously there's a bulk of the data being submitted as sequencing data. Geo does not store the raw data. It doesn't store FASTQ files. It doesn't store sequence files, BAM files. It only stores the summarized data. So it makes it very easy to grab fairly large data sets in fairly short period of time using Geoquery. Geoquery has really one command, so I'm not going to go into any detail. It's get geo. So with an accession, an identifier from this database, Geoquery can go out and grab those data and pull them back in. And just to give you an example of what kind of thing that this facilitates, after your one line, you can you basically have access to the entire an entire data set, and you can quickly jump to, for example, principal components analysis of a cancer data set. The vignette, the workshop materials go through that particular use case in detail. So I'm not going to spend any more time going through those details. The Genomic Data Commons package interfaces with NCI's relatively new genomic data resource called the Genomic Data Commons. The goal in this, in part of the workshop, is to explore somatic variants seen in cutaneous melanoma. The portal.gdc.cancer.gov/repository is essentially a mirror of what we're able to do in Bioconductor. Let me show you. That website looks like this, and you can see on the left side there's a files panel that allows you to do searching and filtering based on files. This is all graphical, which is cool, but we wanted to have high quality, reproducible research, which really involves writing code. So what can we do? Well, I've changed the Genomic Data Commons facetting, the ability to do searching, and converted that into R code. So there's an application programming interface, an API, that allows the genomic data, allows R to interact with the Genomic Data Commons repository, and essentially we're going to take the code here and relate it back to the website. So the second line, starting with fnames equals files, we're going to end up here. This files piece relates directly to selecting from the files in the GDC data portal. We can choose which files by filtering. This is essentially clicking on one of the boxes in the web page. So we're clicking to pull out these particular data types, workflow types, and data formats. This is what it looks like in code. And we're going to grab the identifier, which is going to relate directly to this file name. That's the IDs part. And then we want to add files to cart and download. In R, that's using GDC data. So what you see, fnames equals file, blah, 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 down to GDC data, is essentially what we would do if we clicked on those buttons on a website and then downloaded the data. All of this, of course, is scriptable, and we can find out what data types and things like that are available using the Genomic Data Commons package. So we never actually have to visit the website if we don't want to. And coming back to this idea of data containers, once we have these data, which happen to be in this case somatic variants, we can use the math tools package, which is built for working for with these kinds of things, to do all of these kinds of visualizations and analysis. So in 8 lines of code, we can go and produce, grab data and produce this plot. which is kind of cool. I'm going to stop there and turn things back over to Levi and Ben, and we can answer questions a little bit later.
And I'll be presenting a little bit more on the drug perturbation as well as the radiation perturbation. So really the field of pharmacogenomics is basically generating the data you need for biomarker discovery. So can you predict what are the molecular features that are associated with drug response? And obviously in the context of personalized or precision medicine, this is a very important topic. Many people receive are treated with drugs that have very little effect, or sometimes even have toxic effects and no really therapeutic benefit. So understanding the relationship between complex molecular features of cancer and drug response is really important. The problem, as you'll see, is that despite the fact that a lot of people shared a lot of data, they are very hard to compare because nobody really agreed on different standards, like naming the cell lines a consistent way or naming the drugs a consistent way. Luckily, at the genomic level, there is way more standards that could be used, but it's really at the phenotypic level that a lot of curation needs to happen before you can leverage all those data. And I'll quickly, so we developed Pharmacogex, so I'll quickly present Pharmacogex as our main platform to really implement the curation in a very transparent and reproducible way. So you don't have to, basically all the years we spent curating data, you don't have to do it yourself. You can just start your project with reasonably clean data. And if they're not, you can let us know and we can fix it. And we also complemented this Pharmacogex package with other packages that deal with very specific phenotypes and cancer models. So next. Okay, so as I said, a lot of data sharing occurring in pharmacogenomics, and that's kind of the nice thing about genomics. We've learned how to share data very early on. And as you can see here with a bit of a timeline, you have all the many data sets, including some very famous data sets like NCI60 and the connectivity map, as well as more recent data sets that are getting larger and larger to test more cell lines with more drugs. Next. It's decently used by the community, and really it's really built to save you time. So take a look. I'll describe a little bit more what it does. And we recently had a major update to include summarized experiments in the latest version of our data sets. Next. So the first type of data we have in Pharmacogex is what we draw drug sensitivity data set, where we both have a very comprehensive molecular profiling of the human cancer cell lines. So we have mutation, copy number variation, gene expression, both by microRNA-seq. We have mutation inferred from whole exome sequencing, sometimes whole genome sequencing. We have methylation, sometimes we have also proteomics data from RPP assays. So it's very, very comprehensive molecular profiling. But for each of those cell lines, we also have what we call drug dose response curves, where we basically measure viability with respect to increasing concentration of the drug. And as you can see next, as you can see in the red box, this is really the key phenotype for Pharmacogex, where you have this cell viability decreasing as you increase the drug. That's a nice drug where you can kill more of the cancer cells if you increase the dose. That's what we expect. And if you correlate this molecular profiling with this kind of phenotype, then you can start doing some statistical analysis, machine learning for biomarker discovery. Can you predict from the molecular features whether a new cancer will respond to a given therapy? So there are many functions in Pharmacogex, but basically the first one you would probably like to try is the available p-set. And that will give you a list of all the data sets that are available in Pharmacogex. And in the most recent version, we actually got a DOI for each of those data sets, and they're all uploaded in Zenodo. We call them canonical p-set. They are being processed with what we think are one of the best pipelines. However, there was another presentation during the Bioconductor conference about Orchestra. This is a platform where you can basically request those data sets processed the way you want. We have a larger set of pipelines. You can use Callisto or Salmon. or cufflink, for instance, for gene expression from RNA-seq. You can really tune the pset the way you want, and once you request them, it gets uploaded in Zenodo, and you get a new DOI for your data set. Next. So as I said, this is more about the drug sensitivity data set. And really here, you try to look at genes that are associated. You can go univariate, can go multivariate, but really you try to correlate molecular features with phenotype after the drug treatment. Next. But we also have what we call drug perturbation data sets. And it's really, basically, it contains, this category of data set contains the connectivity map, as well as the L1000, the next generation connectivity map, if you wish, using the cheapest assay. And so what we have here in Pharmacogex is basically a container with the gene expression of the cell lines before and after drug perturbation or drug treatment. And here, instead of looking at association with drug sensitivity, what you look for are genes that are up and down regulated by the treatment, by the drug treatment. And that will give you a little bit more information about what the drug is actually doing. We also have developed another package called ToxicoGX for toxicogenomics, which is basically exactly the same thing, except that the data combine drug sensitivity and drug perturbation in the same container. And it's all about healthy cells. It's about hepatocytes and kidney cells. And really the goal here is not to understand what are the cancers that are being killed by a drug, is really to understand whether a compound will have toxic effects in your liver or your kidney. And so basically it's like the antithesis. You would like to have no change in expression due to drug treatment, rather than finding signatures of, let's say, genotoxicity. Next. And so Pharmacogex is not just about accessing the pharmacoset, which is this container containing all the molecular and phenotypic data. It's also about what you can do, what you can easily do from that new class of object, that new object. So you can easily fit the drug dose response curves, where you increase concentration and measure viability. You have to model it to compute, let's say, the area under the curve, for instance. You can visualize them. We have some nice plotting function in Pharmacogex. You can easily extract the molecular data of interest if you just want, let's say, if you want to summarize the experiment, has a very simple drug versus cell matrix for the phenotype, or gene expression versus cell matrix for the genotype or the transcriptomic data. And then you can use those matrix to do your statistical modeling or machine learning analysis, where you can look at one gene at a time in the package, using the signature functions in the package, where you can do more downstream analysis, or you can apply your own fancy multivariate machine learning analysis based on those simple matrices. So really the goal is to standardize all the cell line drug names, gene annotations, so you can really jump into the machine learning part rather than spending your time on the curation. Next. And so we basically extended that framework to more than drugs. So we went beyond pharmacological perturbation and looked also at radiation perturbation, a field that is sometimes called radiogenomics, where basically it's exactly the same thing, same cell lines being molecularly profiled, but instead of drug response curves, drug dose response curves, what we have here is a radiation response curve, where you have increasing dosage of radiation. On the x-axis, I noted is as gray here, the survival fraction of those cells. So in a sense, it's very similar to Pharmacogex, but the phenotype is very different. It's radiation rather than a drug treatment. Next. Yeah. So yeah, sorry. So the field is less active, so there is only one really big data set that we have created so far, one data set from Cleveland. What they've done is to use the Cancer Cell Line Encyclopedia, the very large collection of molecularly profiled cell lines from the Broad Institute, and irradiate them at multiple doses to generate that very rich data set. We are in the process of creating a more smaller data set that have been published in the literature, but it's definitely a field that is less active in terms of data sharing than Pharmacogex. Next. We have also, we wanted to deal with a phenotype that's slightly different. And here, the cancer models we wanted to analyze were not cancer cell lines, but they were actually mouse models, and more specifically, patient-derived xenografts. So take a piece of tumor from the patient, implant it into mice, and let them grow, and then you can treat those mice with drugs, or you can let them grow as a control. And why we had to develop a new package is basically we have two main reasons. One, the experimental design is very, very different than in vitro pharmacogenomics, but also the phenotype. Here it's not concentration versus viability, it's time versus tumor volume. And so that basically, it requires to have very different modeling functions and the way you store the data. So we've developed a container called Exivaset, which is basically the equivalent of a pharmacoset for those in vivo pharmacogenomics data. And we have one big data set called the PDX Encyclopedia from Novartis, but we also generated smaller Exivaset for each cancer type if you want to analyze those data separately. And we're in the process of including more data sets as well, but this is by far the biggest. Next. Yeah, that's it for my part, and I'll welcome any question at the end of the workshop.
All right, I'm just going to summarize several curated data resources that are selected not only because several of them were developed in my lab, but they are data resources that share use of some core data structures. And the idea behind considering these as a group and using these core data structures is that if you spend a bit of time to learn about these data resources, the summarized experiment, the ranged summarized experiment, the single cell experiment, ragged experiment, and multi-assay experiment, then you will have a common analysis environment and a common set of procedures that will be familiar across a wide range of different kinds of omic data types. And this is really a big difference that distinguishes Bioconductor from the rest of CRAN and from the tidyverse, in that you will see in, I think, most other areas of research, a domination of the table and simple data structures, which has advantages for for a wide range of analyses, but where you want to apply one set of methods to many types of data across different domains. In these domains, we have very complex data. And the problem with using a table type of paradigm, I think, is that you end up having to write a lot of code to do things to these data. And under this paradigm of these core data structures, we have a lot of information about relationships between data types, between data and phenotype data, such that developers can create simple methods that users can apply to quickly analyze these data without writing a lot of code. If you want to learn more about these core data structures, I recommend attending this other workshop, Wednesday morning multi-assay workshop that will include more information about these. So just briefly, these first two are for matrix-like data, such as RNA sequencing, microarray, and single-cell RNA-seq data that are summarized by gene or features like this so that they are matrix-like. Ragged experiment is intended for copy number data, somatic mutations, such as represented in VCF files, and other sort of ragged data where we have different sets of genomic ranges or genomic segments or points per sample. And finally, multi-assay experiment, which is for collections of these different kinds of data for multi-omic studies. So a little pictogram of why to do this. As an example, the Cancer Genome Atlas, here is a schematic of its 19 data types, 33 diseases, and 50 different platforms. And a traditional approach to using files and tables for each of these involves an enormous task of data harmonization and management before you can do any analyses of these. And this approach lets experts like Marcel Ramos do this once so that you don't have to. Marcel is the creator of curated TCGA data and a number of other such curated resources that I'll go over a couple of. So just to put this in context, say of the Genomic Data Commons, which Sean Davis told you a little bit about, this is a very complete resource that offers access to all of the files you can access from the Cancer Genome Atlas as well as from other studies. And also it even offers multiple different data processed through different pipelines. So if you need access to every kind of data or to a type of data that isn't available through one of these simpler, less flexible packages, then you'll need to use the Genomic Data Commons. However, if you don't need the full power of Genomic Data Commons, you may be able to save a fair bit of time and effort by using one of these simpler packages. So TCGA BioLinks is not the work of my lab, but I mentioned because it is an important package. And if you want to get a single omics data type along with clinical data for the Cancer Genome Atlas, this is probably your best bet. And then at the most simple end of this spectrum, again, just for the Cancer Genome Atlas, would be curated TCGA data. And I say it's the most simple because it provides a single object, a multi-assay experiment that can contain the data for all processed data for a single cancer type, or it can harmonize the data across multiple cancer types into one object. And along with the TCGA Utils companion package, it makes it quite straightforward to do a lot of common analyses with the Cancer Genome Atlas. So just to give you an idea of what I'm talking about in code, if you are accessing a data set through TCGA BioLinks, you'll see that it uses the Genomic Data Commons as an engine for doing some of the access of files, but then it provides some additional helpers that load these files and here produces a summarized experiment. But as you can see, you have a number of different choices and a few lines of code in order to get this summarized experiment. Not a big barrier, but you can see the flexibility that it's offering. That here is an example of accessing data for one data type with curated TCGA data. Here it is just one line of code to query what data types are available, one line of code to select those and create a multi-assay experiment. So once you've done one line of code like this, you can move ahead with subsetting by genes, or actually in this case, they're only summarized experiment. There is another helper available in TCGA Utils for adding ranges where possible. So for subsetting by gene, by genomic range, if you convert these to ranged summarized experiments and selecting according to patient characteristics. I wanted to mention CBioPortal Data, which is a very new package just released in the most recent Bioconductor release in April of Bioconductor 3.11. This provides access through an API of gene panels in CBioPortal Data, and there's also another function for accessing full bulk data sets. So this is a new package. Its paper is in press, and it will also be covered in this Wednesday morning workshop. But again, you see the idea is you specify relatively few things and you get a multi-assay experiment as a result. In the realm of microbiome data, we've produced a couple of packages that are worth mentioning. Curated metagenomic data is a manually curated and uniformly processed resource of, it's now over 10,000 samples that are offered through Bioconductor's Experiment Hub. It will be probably over 20,000 by the end of the year. And again, you see here that it is, you have not too much to specify in order to get data sets, in this case, in an expression set format. And then the HMP16S data is a more limited package that just provides access to 16S. This is a type of sequencing data from the Human Microbiome Project that just lists and there's just a single function that doesn't require arguments, but also does help with merging with controlled metadata from dbGaP. So with that, I'm going to move us on now to the workshop. Since this is the very first workshop, this is going to be our first big test of this new system, and this works out well because we have Sean Davis with us, who was the creator of this amazing novel system for offering workshops to all of our participants. So the materials, the full materials for the workshop, if you just want to view them, they're available at this webpage, waldronlab.io/public-data-resources. And I believe the capitalization is important here if you enter this. But I'm going to encourage you to actually run the workshop, and I'll do that myself. So as nervous as I am about live coding, I'm going to give it a try and see what happens. Not exactly live coding, but just to show you how to do it. So I'm going to copy this. I'm going to create a new tab. And please let us know as you try this out. So as you open this up, you're going to see a menu of all of the workshops that are available. So I'm going to choose ours, Public Data Resources and Bioconductor. I'm going to enter an email address, and I'm going to click Submit. That's my email address. Now, this might take a little bit if everyone is doing this at the same time. There's an auto-scaling cluster that has to scale up. One thing I've noticed is that if you're going to run more than one workshop at the same time, it's not a bad idea to create a private browser for doing them so that they're not using the same browser. I'm not sure how necessary this is, but I've heard of a couple of potential issues if you don't do this. So just under File, Private Browser, and then enter this URL. So this is probably taking a little while because everyone is doing it at the same time and the cluster is having to auto scale up. Maybe I can bring you back, Sean, and let you talk a little bit about this system while we scale up.
Actually, if you guys don't mind, just keep talking.
Mm-hmm. Okay.
There's a lot going on here, so.
Okay. So maybe I will mention while I'm at it here that this is a system that will be available during the workshop, but you can also run these workshops at home and on your own computer. Maybe I'll just demonstrate how to do that while we're at it. So I'm going to go to bioc2020.bioconductor.org. And then I'm going to go to workshops. And down at the bottom, there are some instructions here. So this Docker pull, I'm going to get the correct image from the Docker link. Let's see, I want public data resources and Bioconductor. So I'm just going to copy that link. And actually, I'm going to have to change my screen share because I'm just sharing a window, but I need to share my whole screen with you.
Push the launch button.
Oh, let's see if it's ready for me. Oh, mine's not ready yet.
It might be ready even if it's still dark.
Just push it.
Just push it. Ah, okay. Oh, no.
Well, refresh.
Okay. I'll give it a little more time, and maybe in the meantime, I'm going to demonstrate running it locally. I'm asking a lot of my little laptop here, but so there is the Docker URL I got. I'm going to go Docker pull. So this obviously works because I have the Docker client installed. After you go to docker.com and install something, you'll see this little whale icon. That's all you have to do to be able to do this. And then you need a command line.
Levi, there's a couple of people asking if slides are available.
Yeah, I think, oh, look at, no, not ready. I can, maybe one of you can put the link into the chat window to these Google Slides so that they're available immediately.
Yeah.
I can link them then under the files menu as well for people.
All right. So the other thing
I have it up in our studio. Did you want me to do that?
Well, probably at that point I can just bring up, so I can point out each of these. I can just go to the web page as well. If I don't want to do live running of the code, everything is here under the web page. So I've put some links here to the relevant vignettes for packages that are covered here. And then we just have a basic description on the front page. And then there's this button that says workshop, which is the full set of code and output for the entire workshop. So this installation stuff, this is only for if you're running it like on your local R installation. If you're running it through Docker, such as you are, if you go through this cloud instance, mine is ready now. You don't need to install anything. Everything is already installed. So it just takes a couple of minutes because lots of people are starting to use it all at once, and the cluster had to auto scale. So I'm going to go ahead and welcome. What was it? Welcome to BioC 2020.
Yep.
Thank you.
Welcome -to-BioC 2020.
All right, I think I got it. Sure, why not? Save that. Okay, so with a little bit of patience, this is what you will see going through the link to the cloud instance. And by the way, this is also exactly what you will see if you follow these Docker instructions. So if you go through and you want to run this locally using these instructions, this Docker pull, this Docker run, and then going into your browser and clicking localhost 8787, it will look exactly like this, and it will also have exactly all the same software and data installed because it's all within a Docker image. So if you want to play around with one of these, what I suggest doing is finding this vignettes directory. Every workshop has its executable code in this vignettes directory. So I'm going to go ahead and open that. I'm going to skip this stuff at the top. I could knit this whole thing if I wanted to, but what I would actually like to do is just go and run some code. So I'm going to do Control Enter here to run library Geoquery. GSE equals get geo. It's now running the same code that was run to produce what you see on the workshop website. And I'm happy, probably, Sean, you didn't actually intend for me to do a live demo of your brand new system in the very first workshop, but it worked out all right.
So there are 127 instances running right now of this workshop, and there's an additional 100 from other workshops that people have tried. So we're running well over 200 workshops. And sorry about the delay, the auto scaling. So what's going on is that as people ask for instances, if there are not enough compute resources, Google is giving us more. And that takes some time. So I just manually added a bunch of nodes. So about 3.5 minutes of that wait time was just waiting for Google to do its thing. At this point, if people ask for another instance of a different workshop, you should be able to get it in 30 seconds or so. Each workshop gets 7 gig of RAM and about 1.5 CPUs. So it's not a huge instance, but they're not huge, but they're like a laptop basically. And
notably, one of the things I love about this is it's the same machine that they were tested on using GitHub Actions when the authors created them. So we already know that the workshop, anything that's done in here should work, even if it takes a while in here. So I just ran this first line of real code, running get geo on GSE 103512 and selecting the first list element. This is pretty common, I think, the parsing failures, but Geoquery has a lot of problem catching built into it, so it doesn't actually ruin the download. I don't think I have ever run across a geo series that Geoquery could not download. It's very rare. I don't know if it's non-existent, but it's very rare. But the nice thing about doing this compared to just looking at the web page is that I have a running instance, so I can do interactive analyses of this now. I can look at this GSE. I see it's an expression set. I can do things like eData, GSE. And I can adapt this. I can create a new file where I write my own program here. The only thing that I want to remind everyone here is that these are not persistent instances. So after this is, you've been using this for 3 hours, or after it's idle for half an hour, this machine will be destroyed and you'll lose any work that you've done in it. So if you are actually writing a program that has some code that you want to keep in it, then save it elsewhere. So maybe we should open up the floor now for some questions. Now that this is working, I can keep on running through some of this, but I think we only have about 5 minutes left. It would be a good time to take some questions and also any feedback about people running a workshop.
So Levi, if I may, I forgot to mention this. There is a poster about ToxicoGX during the Bioconductor 2020 conference. There is also a specific workshop on Xiva, Pharmacogex, and RadioGX presented by Peter Smirnov, Christopher Eels, and Arvind Singh Mer. I'm trying to find them in the agenda, but if you want to go deeper into those packages, you will have dedicated workshop to do so.
Thank you, Ben. And Mikhail, if you have your eyes on the questions, we could start taking questions now.
Well, there are several questions, and one of the most popular is, what would be the single-cell RNA-seq data sets that are available?
Right.
The package.
So I wish we had here. Sorry, go ahead, John.
Go ahead. No, go ahead.
I was just going to say we need Stephanie here.
Well, there's the Conquer project from the Mark.
Right.
Who's the guy in Lausanne?
Mark Robinson and
Mark
Charlotte
Robinson.
Sonesson.
A very large online system for that. Yeah.
There's a great experiment hub package called scRNA-seq that has curated data sets.
So I'll write those out here, scRNA-seq and Conquer. These are a couple of recommended ones. Conquer, by the way, uses a summarized experiment to provide gene level and transcript level summaries of every data set, which I liked. I'm not actually familiar with scRNA-seq.
Sean, does the awesome single cell resource list data sets too?
Not so many. It does have data portals and collections in it. Unlike NCBI, or unlike other data types where NCBI and Ensemble EBI have been sort of the leaders, there are a number of very large single cell data sets that are available in private portals. The Human Cell Atlas, for example, from Chan Zuckerberg. So those would be listed as resources.
Another question. So Levi, you created a lot of packages for different types of omics data. And obviously there are other types of omics data like proteomics, single cell proteomics. Do you have plans to make like a unified structure that can handle all those multi-omics data sets?
Well, I believe the uniform structure exists. That is multi-assay experiment. And as far as the work remaining to be done is to harmonize those data sets. And we're doing we're doing what we can. Our current efforts now are focused on single cell multimodal data sets and harmonizing those as multi-assay experiment. And also CBio portal has a variety of different kinds of omic data sets, including proteomics. I should also mention that Laurent Gatto is creating new data structures specifically for proteomics and data sets where you have proteomics and genomics together. Also, by extending multi-assay experiment, but with some special things relating to peptides that will make those data more usable.
And another, yeah, that's good. Another question is, are there any resources for accessing long-read sequence data?
Geoquery. I don't know of anything specifically for long read sequencing data. I'm sure that they exist in the Gene Expression Omnibus, but I don't myself know of specific data resources for that.
We didn't talk about it today just for time, but we have a separate resource called Omic IDX or Omic Index that allows you to do searching against the sequence read archive, SRA. And one of the tags that's included in that resource is the sequencer and the sequencer model. So if you wanted to search for specific sequencer models, for example, if we're looking for long read sequencing, you can do filtering based on that kind of parameter, in addition to all of the usual searching for keywords, things like that.
So Sean, I know Omic IDX will give you all of the sequence read archive accessions. Will it also give accessions for Geo if the process data have been put in Geo?
Yes, so there are cross links between Geo, SRA, and biosample. And for any sequence data in NCBI Geo, the actual data will be in the sequence read archive. The opposite is not true. So the data in SRA are not always included in Geo. The process data are not always included in Geo. And yes, Omic IDX can search those three groups: biosample, Geo, and SRA.
I want to say to everyone here that Omic IDX is amazing. It's a web API that you can use through a program or just directly in the web browser manually that searches all of the sequence read archive with a number of different options instantly. It's amazing. Without installing anything, you type something like you're interested in a certain sequencing platform and you get all the results instantly.
Just for the record, is this URL that I'm pasting in the chat window, that's what you mean?
Let's see.
Omics IDX.
I don't actually see the chat window.
It's github.com omics IDX, omics IDX.
Yeah, I don't think that's it.
No. I guess, Sean, you would be, is it your repository?
Yeah, let me.
Actually, when you go to the GitHub Omics IDX, it actually says that the current REST API location is API Omics IDX cancerdatascience.org. Is that correct, Sean?
Yep.
No. Okay, so yeah, if you go to GitHub Omics IDX, it provides the link to where you need to go to.
Do we have any more questions, Mikhail, or shall we let folks get to the next session?
The other questions is pretty much cover what we already talked about. What are other data sets, whether you can access ChIP-seq data with Geoquery?
Yes, in some cases. As you can imagine, Geo was originally written for two-dimensional data, so Excel spreadsheet kind of data. So if the depositor has submitted, for example, genomic regions by counts as a matrix, then those data might be available as a get geo type query. Geoquery can also grab all available supplemental files, and what you'll often find for ChIP-seq data is that the data sets are available as supplemental files, sometimes WIG files, sometimes BED files, sometimes BAM files. So those require a couple extra steps, obviously, to get them into something usable in Bioconductor. But we have loaders for all of those kinds of data. So generally, if there's a supplemental file available, Bioconductor or R can deal with it.
Right, we have to stop here. Well, it's more than 200 people. So thanks, everybody.